Hey team, I hope you're all doing well today. I've had a lot of requests for some neuroscience videos. So I wanted to come in today and talk a little bit about neurons. Um, and well, that should be a capital letter. Uh, so the brain has lots of different kinds of cells, but kind of the workhorse cell, or the cell that makes everything work, is called neurons. And they are responsible for basically carrying information around the brain. And they're really, really cool, and they have this really weird, funky shape. If you've ever seen a medieval mace, or it's technically not a mace, it's a morning star, it looks kind of like that. But it has the middle of the cell has a really, really long body. And then on one end, it's kind of got these wavy tentacle things. And on this end, there's an actual body of the cell, and it's also got some wavy tentacle things. Okay, and so we call one end, here's the cell body, right? This is like where like the nucleus and stuff would be. And then we call the middle of the cell here, this part right here, we call it the axon. And then down here, there's a lot of names for this thing, but I'm going to call it the terminal. That's not how you spell terminal. And these little wiggly things that are sticking off here are called dendrites. And basically, information flows through neurons in one direction. So info, what color should we use for information? What's a good information color? I feel like blue is a good information color. So information is always going to enter the cell here, move through the cell in this direction, and leave the cell here. Okay, so we have info is going to enter the cell here, and information is going to leave the cell here. And so that's sort of important because these cells are what carry information from one part of your brain to the other, right? There's a part of your brain that processes vision, and so signals go into that vision part of that brain, but just seeing a cat doesn't really do much for you if you're a living, thinking human brain. You need to carry that information to somewhere that can understand what a cat is and the word for cat, and those pieces of information are all in different parts of the brain, and these cells are going to carry that information around. And there's kind of a cool mechanism for how that works. And if you've ever heard about something called neurotransmitters, that's what kind of makes this whole process function. So we have to remember that our brains are floating in a liquid that's basically just salt water, right? And so it's full of this chemical called sodium. Okay, and I'm going to draw Na as the chemical abbreviation for sodium, and you see I'm drawing little pluses next to these sodiums because they have positive charge. I'm going to draw a few that look like that, but I'm just going to draw the rest of them as circles for simplicity's sake. So there is sodium all around this bad boy here. Okay, so this guy's floating in sodium, which means that the outside of the cell with all the sodium is super duper positive. Okay, and so when we think, if you've heard that cells, that your brain kind of works based on electricity, or there's electricity moving through the brain, that's what they mean. People don't mean electricity in the conventional sense with like electrons floating through a wire. They mean electrical in the physics sense, right? Where there's an electrical gradient, right? You can think of like a magnet, right? These guys are all really, really positive. So the outside of the cell is more positive than the inside of the cell. Okay, so outside of the cell has a lot of sodium. Inside of the cell has less sodium. And if you're like an expert in neuroscience, you're about to tell me right here, Bortz, there's like a big thing about potassium that you're leaving out here, and you're right, I am leaving out this big thing about potassium, but the intended audience for this is like people who just want to learn the basics of science or high schoolers. So if you want to learn the technical aspects of this, go watch a video with the technical aspects. So what happens here is there's these chemicals called a neurotransmitter, and they're going to come from another neuron, and we'll get to that in a second. But these neurotransmitters bind on to these dendrites. We'll make a little key down here. The purple circle is going to be a neurotransmitter. We'll make the little red circles are going to be sodium. And so what happens when these neurotransmitters bind to the dendrites is if we kind of zoom in a little bit on one of these dendrites, in the cell membrane of one of these dendrites here, there is so if we think this is the cell membrane, there's a gate that's closed. And when a neurotransmitter binds to that gate, it's going to open up. So one of these neurotransmitters, what they do is they bind to this gate here, and they're kind of like the key. They open it up. And as you know, due to the properties of diffusion, when there's more sodium outside the cell than inside the cell, all of these places where the neurotransmitters have bound are going to let the sodium move into the cell. And that's going to move these sodium ions into the cell. And that's going to change the voltage of the cell, right? It's going to make it more positive, okay? And so these sodium ions are going to get collected up in here, and they're going to keep moving in. And eventually, there's another gate. There's another kind of cell gate here. Right here in the neuron. And that gate won't open until this part of the cell is positive enough. Okay? So this guy only opens at a particular electrical voltage. And it's different for every neuron. But when that voltage is reached, this guy is going to move and open up, and that's going to let sodium also move down the middle of the cell here. So we'll kind of put some sodium. And because we want this to happen really, really fast, there's also little channels embedded in this part of the membrane. And they're also voltage gated. So as the sodium comes into contact with these channels, they're also going to open up and let sodium diffuse in through even faster. So we'll get more and more in sodium coming through. So as the signal moves down the neuron, the signal goes faster and faster and faster, because with each of these gates that gets opened up, more and more sodium gets let through. And eventually, when this guy fills up with enough sodium, the voltage of this guy will change. And so down at the end of this little guy here, there's these little neurotransmitters that are just waiting here. They're just chilling. They're not doing anything. And when there gets to be enough sodium at the end, some more voltage-gated channels are going to open up. And that's going to allow these neurotransmitters to leave this neuron. And what they're going to do, these neurotransmitters are going to go and they're going to find the dendrites back here of another cell. And they're going to contact that cell, and this whole process is going to start over again. So what you end up with, and I'm just going to kind of draw this simpler. You can imagine this as kind of be the cell body, and this is the terminal. So what you have is these chains of neurotransmitters where the these chains of neurons where the neurotransmitters are jumping from this one, and they activate this one, and that carries sodium down this one, which activates this one. And that carries signals through big parts of your brain. And so you might be asking me, well, that's really great, and that's really cool, and that explains how a signal gets through my brain once. But hey, every time I use a part of my brain, don't I have to be able to reuse it? Isn't this guy full of sodium now? What are we going to do about that? And so what happens is there's these little things in here. Maybe we'll put these in a different color. There's these little proteins embedded in the membrane, and they're called sodium-potassium pumps. And we're not going to talk about potassium in this video. But basically what the cell does is it uses energy to work against the laws of diffusion. And what these sodium pumps are going to do is they're going to pull the sodium. They're going to use energy, right? And anywhere where there's a sodium-potassium pump, they're going to use energy to pull sodium out of the cell, which is going to make the cell more negatively charged overall. Okay, so it's this difference in electrical gradients, which is what allows the cell to carry information, right? So what we see is the cell becomes more positive, positive, positive. The first wave of positivity opens up a bunch more channels, which lets the cell get even more positive. And then as the cell reaches its threshold for positivity, it releases a bunch of neurotransmitters. And then these pumps pull the sodium out until the cell gets negative enough, and all these gates close back up. And eventually, when neurotransmitters hit the dendrite of those cells again, they'll open those channels back up and start the process over, right? So we have this kind of process where we use electrical signals to make the cell positive enough, which releases the neurotransmitters. And then we pump the sodium out of the cell, which makes it more negative. And then when the cell gets activated, it becomes positive again, so on and so forth. Which leads us to this signal, right, where the neuron is either firing or it's not, right? The neuron doesn't fire any more or less. It just either releases its neurotransmitters or it doesn't release its neurotransmitters. Hopefully that made sense to you. We're going to do some more videos in the future where we talk about this a little bit more. This is just kind of the basic information you need to know before we talk about the cool parts of neuroscience. If you have any questions, drop them in the comments. Feel free to shoot me an email. I hope you're having a great day. May the force be with you.